Thank you so much, everybody, for tuning in. I am thrilled to introduce you to someone who you may already know, Dr. Steve Hsu, who is a physicist and a professor at Michigan State University and the founder of an organization called Genomic Prediction. And we're here to talk about clustered regularly interspaced palindromic repeats, of course. In other words, CRISPR, and more generally speaking, genetic testing and genetic editing, which, as I was mentioning to you, Steve, right when we got on, is truly one of the most fascinating threads of science to me, and perhaps one that is also most volatile is the word that comes to my mind. And just what I mean by that is the potential seems like it could go in extreme outcomes. So I think it's really important to be having conversations about it, just about how it works, what's possible, the ethics around it. So let me just let you add any detail you'd like to the introduction of who you are.
Great. Well, Nick, I'm very happy to be on your show. I would say that, first of all, I should clarify that although my background is in theoretical physics, which people find strange since most of what we're going to talk about, I think, on the show is genomics, genetic engineering. I've been working in the field of computational genomics for about a decade now. And what got me interested was the promise of reduction in cost of genotyping, or reduction in cost of reading out people's DNA from a sample, say from some saliva or some blood or even a skin cell. And 10 years ago, I just did some what physicists call back of the envelope calculations. They're just sort of simple calculations or estimates for, gee, what would it be like if the cost to genotype somebody fell below $100 and consequently we had millions of genomes to analyze. And the conclusion that I came to even back then was that once we had that level of data, that amount of data, we would be able to build mathematical models that actually connect the state of an individual's DNA to the individual differences. So in the field of psychology, you guys would call them, I think, individual differences, the ways in which people are different from each other. So why does somebody have blue eyes and not brown eyes? Why is somebody 6'4" and not 5'4"? Why is somebody really susceptible to breast cancer and someone else isn't? Those are all individual differences in, the technical term here is phenotype. And we know from studies where we look at, say, twins or families or kids adopted into families, the degree to which those individual differences are determined by the genetic differences. So in other words, identical twins are much more similar on traits that are highly heritable, highly controlled by DNA, because they have the same DNA effectively. Whereas a kid, for example, who's adopted into your family and doesn't share very much in the way of genetics with, say, your brother and sister, will tend to be less similar than you and your sister are on a lot of these traits. So already a lot was known about the general level to which certain traits are heritable or not heritable. And what the mathematics told me 10 years ago, and it turned out to be true, is the degree to which we would actually be able to predict based on the DNA certain aspects of the individual. And hence the name of the company that we started is actually Genomic Prediction. And that's actually a technical term in computational genomics. It refers to the ability to analyze a bunch of data and build a mathematical predictor or an algorithm that, given the DNA of an individual, then predicts something about that individual. So that's the very specific area that I work in. And it's actually a natural extension of the previous work I had done, both in computation and also in theoretical physics. So people think it's like a very weird departure that a physicist would be working on genomics, but actually it's not, because modern day genomics is highly, highly computational. And I should qualify that I'm not a molecular biologist. So I'm not a guy who has actually ever worked with CRISPR directly. And so what I know about CRISPR is, I suppose, as an informed scientist. However, I'm not, there are many other people who are perhaps more knowledgeable on the specifics of the molecular biochemistry. Now, the aspect of CRISPR that I think I'm probably better informed about than most people who really work on CRISPR is the extent to which we will know in the next 5-10 years exactly which edits we want to make. So you have to solve my problem first, which figures out which aspects of the DNA of an individual. There are about 3 billion base pairs of information in your genome. You need to know which base pairs you want to edit and what the consequences of those edits are before you can apply even the most science fiction-y, whiz-bangy CRISPR technology. So the two problems are very much related. I'm more on the computational side.
Yes, makes perfect sense. And it's not easy to have a conversation like this when you don't know exactly who the audience is, of course. And so my assumption is that there's a mixture of people who are listening and watching now who are completely unaware of CRISPR, what we're talking about, and don't really know about what's going on in gene editing or genomic prediction. And then there are some people who have read and watched up on it and listened to many and know a lot about it. And so I want to make the decision not to go into detail about the precise mechanisms by which CRISPR works, and because one, some people might already understand it and I don't want to be redundant, and two, if you don't understand it, I don't know that this is the best format to provide a complete explanation. And I would recommend people to watch the documentary that you're featured in called Human Nature. I've watched every documentary I can find on CRISPR and read a lot on it as well. And this one is particularly good at explaining how it works. But to give a simplistic analogy that people often give, we now have the ability to essentially edit the human genome. And the analogy that people often give is that you can have a Word document and with many words written onto it, many sentences, many paragraphs. And CRISPR is essentially the equivalent of being able to go in, place your cursor wherever you like, delete words, edit the spelling of words, or potentially even add words into it. And again, there are very specific mechanisms by which this works. But yeah, essentially we have, a protein called Cas9 that can be programmed, which can cut and edit DNA sequences. So, again, I know it's not easy because you don't know how far in depth to go with an explanation, but is there anything else on the level of a simplistic explanation that you'd like to add?
I think for the lay person or the the person who's not an expert in molecular biology, the way to think about it is that each of us has a genome. There's information in that genome. It turns out there's about 3 billion individual base pairs, as I mentioned before. And what makes two people different from each other is typically we differ at maybe 1 per 1,000 base pairs. So only a few million out of those 3 billion locations would say Nick, you and I differ at. So there are specific places where our DNA is different, and then there are huge chunks where it's basically the same. And one could just imagine that these wizards in the lab have developed a tool that they can inject into a cell of your body. It goes into the nucleus of the cell where the DNA is and edits the DNA, and it makes the changes, whatever changes are desired. You can design the molecule to make specific cuts or changes to an individual's DNA that are desired. And of course, there's much more detail and subtlety there. There's an error rate. Sometimes the thing edits the wrong site. Sometimes it doesn't work right. But they're improving it. Year by year, it gets better and better, more accurate, more effective. And so I think it's not a crazy at all extrapolation to say that we'll soon have extremely accurate, extremely safe gene editing technology. It is already being used today to cure diseases like sickle cell anemia, et cetera. We can get more into that, and it's certainly in the documentary. I also want to recommend the documentary. even though I don't particularly like the way they portrayed me in the documentary, I do think it's an excellent documentary. And at the beginning, when they explain the sort of molecular dynamics of how CRISPR works, it's extremely well done, and the graphics are beautiful, and everything is beautiful about the way they explain that. So as a kind of scientific educational introduction to CRISPR, I agree with you. There's nothing better than the first, hour of Human Nature.
So many directions we can go with this, but one thing that I think is worth saying here in the beginning too is there's kind of two topics here that we're addressing. One is genetic testing, advanced genetic testing, and then the other one is genetic editing, and those are very different. I think those sit differently with people, because genetic testing is simply gathering information, and I don't know that anyone would make an argument that there's anything unethical about genetic testing. Perhaps you've heard arguments though, but I was very intrigued by something that I understand you do at Genomic Prediction, which is, you have two parents who want to conceive a child, and you help them conceive by basically doing in vitro fertilization with gametes from each of them, and then you'll basically create up to 10 or so viable embryos, which then you can, and of course, as people may or may not know, each embryo will have different DNA. They're pulling different DNA chromosomes from each parent. So each of those 10 embryos, although they're from the same two parents, will have a different genetic profile, which you can see in pretty significant detail. So then the parents can technically choose which embryo to implant. Is that correct?
Yes. Do you mind if I just give a short intro to your audience on this?
Please.
So there are two aspects of this. One is the whole area of in vitro fertilization, which predates our company. Our company is a young technology startup, whereas the oldest person who was born through in vitro fertilization is now in her 40s. So that's an older technology. It's been around a long time. Currently, in some countries, like for example, Denmark, about 10% of all babies in Denmark today are born through IVF. In most developed countries, the typical number would be something like 5%, 3 to 5% maybe. And so that's a substantial number of kids. That means if you go to a playground, if you go to a kindergarten or a schoolyard, there will be multiples of IVF kids running around. So there are many families that wouldn't have a kid if it weren't for IVF. And it's typically applied in cases where there's some fertility issue. Typically, a very common situation would be the mother is a little bit older, and women's fertility tends to fall off actually much earlier than people think. It actually starts falling off in their 30s. And so already a woman who gets married and has a career, and at 35 is ready to have her first kid, it would be very common for that woman to, for example, need IVF. So that's a very widespread technology. Obviously, 10% of the babies in some countries are being born through IVF. It's widespread. People are using it all around the world. And I think by and large, everyone agrees it's a super positive development to be able to do this. Many people have benefited from, many people exist today who wouldn't have existed otherwise. But when IVF was first developed, it was extremely controversial. And the original scientists who developed the technique, and a couple of them went on to win the Nobel Prize in medicine for it, they were reviled as monsters at the beginning. So they called these kids test tube babies. I'm older than you probably. So I remember watching as a kid on television. I think I was having breakfast with my family as a kid, and on TV, on CBS News, they were talking about the first test tube baby, and it was a huge deal. It was a sensation. So kind of similar to like CRISPR now or something like that now, but it's become normalized. And the lesson I'm trying to draw from that is that it could be a radical technology that, wow, this baby was conceived in a test tube, artificially outside of the mother. And yet it's become normalized. It's become so normalized that we all have friends who maybe we don't know it, but they had their kids through IVF. Now, as you mentioned, so the typical process in IVF it's not uncommon for the family as part of the process to have 5 or 10 or even 20 fertilized embryos. So after the sperm meets the egg, the egg is fertilized and then it starts to grow. And at that point, it becomes an embryo. And typically, standard practice today is you let the embryo grow until it's about 100 cells. So it's like a little soccer ball, and then you freeze it. And typically what they do before they freeze it is they take a few cells off the embryo, typically from the region that is going to grow into the placenta. So not from the region that is going to become the kid, but from the region that's going to become the placenta. And but those cells have the same DNA as the kid will have. And from those few cells, we can amplify the DNA enough to get the whole genome, information about the genome of each individual embryo. So it is now standard, it's very common for the embryos to all be genotyped, at least at some level, before the parents make the decision which of these 10 embryos, I'm just using that as an arbitrary number, which of these 10 embryos is going to become my son or my daughter. And the additional information that they have, thanks to this genotyping, allows them to make better decisions. And so that's what genomic prediction does. We both have a special process for the DNA amplification and the genotyping, but also for the computer processing of that information. So we can tell them, well, all your embryos look healthy. However, number 4 is super high risk for breast cancer. And we know you have a family history of breast cancer, and you have told us that you wish to implant an embryo that is in the normal range for risk, not an extreme outlier for high risk of breast cancer. And in that way, the families are able to help ensure that they have a healthy child, even if, for example, they have a family history of disease. The most common use of genotyping for embryos is just to check for Down syndrome, to check that the chromosome count is correct. And if you've had a kid recently, just in a normal pregnancy, you know that there are now techniques that just from the circulating blood of the mother while she's pregnant, we're able to determine whether not the embryo, in this case, the baby, the fetus, we're able to determine whether it has an extra chromosome and therefore will be a Down's baby. So screening for Down syndrome is very common both in IVF and in just regular pregnancies, just because of the advancement in genetic technology.
Right.
The envelope for all this is, for example, we can, from that DNA sample that's taken from each embryo, we can predict their eye color, their hair color, how tall they will be. We can say certain things about some psychological traits or even intelligence. All of the things I just mentioned, we actually do not offer for ethical reasons. We do not offer them. We only offer in our report information about health risks, so things related to well-defined disease conditions. But what is controversial, of course, is, for example, the idea that someday we might offer screening for intelligence, or maybe even CRISPR editing for intelligence, things like that. Those are the things that, for example, I think, I don't recall exactly what kinds of questions they asked me in the documentary Human Nature, but I think they were really focused on that kind of thing because it's the most sensational.
Right.
But much less sensational would be, and this is an actual use case that we often see, is the family has a history of breast cancer. They want to make sure that the embryo they implant, they're not trying to create a super baby. They just want to make sure that the embryo they implant is at least in the normal range, not 99.5 percentile for breast cancer risk, which would be not uncommon for people maybe in their family because of their particular genetics and history. They just don't want that outcome. They're not trying to get a super baby, but they're just trying to screen against a really negative outcome. And that is what we do.
Fascinating. And you said it, you used an interesting phrase, which is that people can make better decisions. And it's interesting when you really start to think about how that could be interpreted. And you've just spoken to this, because better decisions could mean simply screening out a genetic condition or that puts you at risk, or that is definitely going to be the case, like you definitely have the gene for sickle cell anemia, for example. But then, of course, some people will interpret better decisions in these other ways that you alluded to that could sort of lead to what everyone likes to describe as designer babies. So I find it very interesting, and I didn't know that in genomic prediction, you can access that information about eye color and height and things like that, and yet you don't include that in the report for ethical reasons. That's fascinating. And is it true that some people in some places do provide all of that information for people and do allow people to choose what eye color their baby will have?
It is said that, so there are some IVF clinics that have said they will offer things like selection for eye color. Some of the more complicated traits like height or cognitive ability are things that only we can do. So I think that actually isn't possible for other entities to do it, since I don't think they have the capability. Although they might wrongly advertise it, but they as far as I can tell, they can't do it. I should say that, by the way, this is a very, as I mentioned before, there are many people doing IVF around the world. Millions of embryos are genetically screened each year around the world, and I think we've tested embryos for something like 200 different IVF clinics around the world at Genomic Prediction. So it's a very big, sort of sprawling, widespread human activity by now. Very different from the first instance back over 40 years ago.
Right. So what I understand you saying is that essentially, with the computational power that you all have developed, you're really at the cutting edge. You're kind of in the lead in terms of what you can predict, which to me gives you a lot of potential influence. So a question I'd be curious to hear you answer is, what factors are you considering when you decide on the ethics of whether to provide that type of information?
I think the easiest thing for us is to just very gradually extend what was already standard practice in IVF before we founded the company. So before we came on the scene with our more advanced technology, what was already being done was screening against Down syndrome. That's a very gross genetic change because you have an extra copy of a chromosome. So it's very easy to detect, and a lot of the older technologies could do that. You didn't require really the fanciest stuff. The other thing that was already very common was screening against what we call monogenic conditions. So in other words, sickle cell anemia results from just one single genetic change. And so if you zero in on that, like say there's some reason to suspect, say it's an African-American family, and you suspect there could be some risk of them being carriers of that mutation, you can zero in and just look, is that specific change one letter of the DNA alphabet? Is it there? And if it's there, can we use one of the embryos where it's not there? And that was already widespread, and it was widespread for sickle cell anemia. There's a set of mutations called BRCA, which drastically increase breast cancer risk. There are other known mutations that do really terrible things like cystic fibrosis and Tay-Sachs, very exotic diseases that maybe only affect 1 in 1,000 people or 1 in 10,000 people, but they're well understood. And it's a very small move from screening against the breast cancer BRCA single gene mutation to being able to figure out, as we have and other researchers, that there are lots of ways in which you can be high risk for breast cancer. That is just the easiest one in terms of its genetic architecture. Just one change makes you super high risk for breast cancer. What we've uncovered is thousands of individual variants in the genome, where if you just happen to have an excess of the risk promoting alleles in those different places, you could have equivalent breast cancer risk to someone with the single BRCA mutation, but it's just that the architecture is more complicated. The way that the risk arises is more complicated. And until we came along, you could not detect that. But now we can detect it. And there are about 10 times as many women walking around today who are at super high risk for breast cancer because of the polygenic mechanisms that we can detect than are carrying the well-known BRCA variant. So it's a much larger addressable population that we can detect from their DNA that they're high risk for breast cancer. And similarly, in the embryo situation, most of the time, if a family comes to us and they say, we have a history of breast cancer in our family, we can run a screen on them and determine, no, you're not actually carriers of BRCA. It's much more likely that even though they have this family history, they are not carriers of BRCA, but they just have, they have this polygenic risk spread throughout their genome. And that's the sort of new situation. So the benefit that we're offering that family is no different from the benefit that they were being offered before by the older technologies that could only detect BRCA. The benefit is we'll give you a normal risk baby instead of a super high risk baby. The difference is just that we're able to detect a larger subset of the situations in which their genetics are giving them that extra risk. So the moral question in that case was almost not there at all, because it was almost as if you just had a better way of detecting Down syndrome, or you have a better way of detecting breast cancer risk. And all of those are pretty much uncontroversial, at least within the IVF industry, for sure. There was just no question. People would just, if you're a doctor or a genetic counselor and you're trying to help this family, you don't even think twice. If somebody comes to you and says, hey, we'll determine this family has a history of heart attacks, and a lot of the men in the family die before they're 45 or something of a unforeseen heart attack. Can you screen for that? And it turns out we can screen for that. And there was never any ethical discussion over whether that was acceptable or not acceptable. The places where people get more uncomfortable are, of course, cosmetic traits like eye color or height, and actually skin color. So in some countries, like for example India, they really like to have lighter skin, especially girls. And so there's very strong demand for, selecting a baby or an embryo that's going to have lighter skin. And we can do that, but we do not do that. So that's one where we drew the line. And of course, the biggest one that people are concerned about is cognitive ability. Like, can you tell me which of these embryos is going to have the highest IQ or something like that? And we don't do that.
What would you say if someone were to push back and say, what right do you have to withhold information about my embryo?
It's a great question. And we have extensive conversations within the company, often with different, viewpoints on where to draw the line. For example, we have some ability to predict autism risk. And many parents today are concerned about autism, but the predictor that we have is still not as strong as some of the other predictors that we have for risk. For example, we have a very strong schizophrenia risk predictor. And so, we have not rolled it out. We have not rolled out autism risk in our report. We may do it at some point, maybe after the predictor gets a little bit better. But we have very difficult conversations, even within the company, as to exactly we've now developed through this analysis, we now have an autism predictor, but it's got these characteristics in terms of how good it is. Should we use it or should we not use it? And so there's no simple answer to these questions. And one of the reasons I go on this show or in the documentary Human Nature and other documentaries, even though I know you seem like a pretty fair, and actually you seem like a really honest person, Nick. But sometimes the director of the documentary isn't very honest, and they say, oh, we're going to give you a fair shake. But then they spend the entire time talking about IQ, and then they cut everything out except me answering some question about IQ or something like that. You can imagine what it's like. But I still go on these shows because the technology is not going to be put back in the bottle. Okay. After World War II, atomic weapons, we didn't just put them away. Okay. Every country was racing to get atomic weapons. Okay. And neither CRISPR nor these computational genomic advances are going to be put back in the bottle. And so the important thing, from my perspective, thinking of the long term, not the near term interests of a company or something like that, but the long term interest of the human species really, is for us to discuss all of the things that you and I are discussing now, and then come to some societal conclusion. Like, okay, if you could imagine a society saying, well, if the embryo looks like it's at super high risk for intellectual disability, the company should be allowed to flag it for the parents the way that they might flag breast cancer risk. And I can imagine a just, kind, thoughtful society coming to that conclusion. But I can also imagine a just, kind, thoughtful society saying, that's wrong, we don't want that. And I don't even know what the right answer is. I wouldn't impose my views on this on a society or a country. But I think the first step to getting to a conclusion, passing the right laws or regulation, whatever it is for first people to be informed about what is actually possible. Most of what you and I are discussing is not known, is not understood by 99.9% of politicians, national leaders, et cetera, et cetera. It's only a few scientists and practitioners, IVF doctors, embryologists who know this kind of stuff. Most people don't. So that's my perspective on it.
So there's a few things I want to kind of lean into here. And first, I just want to clarify too for watchers and listeners that we're not even talking about genetic editing yet. And maybe that's clear to people, but we're still just talking about genetic testing and what we can know and predict. And one thing I hear you saying, go ahead, please.
Nick, if I could just emphasize, just to reinforce what you said, not only are we not talking about science fiction-y editing of genomes, we're talking about screening of embryos that is happening in every major city in the world. In other words, if you're in the United States and your town has a population of 200,000 people or 100,000 people, there's an IVF clinic in your town, which is producing embryos for families that hope to have a child, and those embryos are being genetically screened. And so we're talking about decisions that affect every single part of the country and the world. It's not hypothetical in the slightest.
Right. And essentially, your company is just becoming capable of screening to a level of detail that's really unprecedented. So congratulations for that success, by the way. And okay, yeah. So one thing I hear you saying is that part of the ethical considerations when it comes to telling parents what these predictions are, what a phenotype might be, is the certainty you have about whether that is accurate. Because of course you wouldn't want to be only somewhat certain that there's going to be an intellectual disability and then tell them that, and then not choose that embryo, but you weren't sure anyway. So that's interesting. And then the other thing I wanted to bring up is, your quote from Human Nature that really stuck with me, which was so good. I'll quote you. You said, "Sex is for recreation, science is for procreation." And I think, what we're talking about right now is what was behind that statement for you, that we can be scientific about the way in which we reproduce, and by using this type of technology. And so, and the other thing I wanted to raise too, that which you also talked about in the documentary, is the idea of eugenics, which of course is even just a word that is very aversive to people. But if you break that down to mean, it means good genes. And the reason that word is so aversive to people, of course, is because the history of eugenics. But it's not, I'm with you that it's not necessarily controversial even to say that we would like to improve the human gene pool. And that's essentially what is at the heart of eugenics. But of course, the means by which we try to achieve that are very dicey.
I mean, Nick, you're a highly educated, thoughtful person. So for you to say it's not crazy for us to talk, in some caring, thoughtful way about, quote, improving the human gene pool. But many people would say, when you say improving, you're making value judgments. You're saying this person is better than this person, and it's just intrinsically wrong. And I understand that perspective, and I understand, if you disregard that perspective, what it could lead to, like in Nazi Germany or something like that.
Right.
But on the other hand, if we come back to some of these situations like Down syndrome, or some of these mutations lead to a child who is going to die right away, or maybe live 10 years and die. Those are horrible, horrible situations. And yes, in a way, we are saying it is better for them to have an embryo without that mutation than to have the embryo with that mutation. We are making that value judgment, but we want to be extremely careful whenever we do that kind of, comparison of individual, people or genomes. And so I understand all caution should be exercised about this stuff. But on the other hand, if I can help a family have a healthy child that they otherwise wouldn't be able to have, it's almost the same as a doctor curing a sick child rather than letting the child die, right? And so it's one of these places where if you could write 100 PhD dissertations on bioethics or philosophy related to what we do every day. So yes, it's quite a nexus of things right now.
And just to, complicate this conversation about value judgments even further, one thing that often comes to my mind, on this is because, like you said, Down syndrome is very easily recognized. It's an entire third chromosome on the 21st pair. And we screen for that. But one thing that always comes to my mind is my father has spent a career in special education. And he primarily worked with people with Down syndrome and still does. And I grew up knowing people with Down syndrome very well, actually. And as you will, and as everyone probably knows, who knows anyone who has Down syndrome, or the parents or teachers or anyone, there is definitely undeniable beauty in that condition. People always speak to like this uninhibited joy, granted severe intellectual limitations, but there's a sort of collateral beauty in it. So even with something like Down syndrome, which if you said you are going to have a baby with Down syndrome, that would hit someone almost as like tragic news, but often people turn out to say like, I wouldn't have it any other way once that child becomes an adult. So like even with something that might initially come across as like objectively wanting to avoid that, even that's very complicated. There are even value judgments in that, but there has to be some set of conditions where it's just objective. No one wants a baby who's going to die in a week, period. And there are genetic markers that can predict that. So certainly there's somewhere where we're all going to agree, but soon it gets gray, and the gray area is big.
Yeah, I do want to point out that in the case of Down syndrome, there are people who campaign against, if they had their choice, they would make it illegal to screen against Down syndrome, or for example, to abort a baby, or I'm sorry, a fetus, who is found to, likely be a Down's baby. And so even there are tremendous disagreements. I grew up, when I grew up in Ames, Iowa, my next door neighbor, who was adopted, our next door neighbors adopted a son, and that son was, the word that we used then was retarded. I don't think he used that word anymore. He had an intellectual disability. He was not a Down's baby, but he had severe intellectual disability. And nevertheless, he and I were friends, and we used to play together all the time. The mother and father were lovely people, very devout Christians, and the mother was a school teacher. So they had a lovely family, but I could just see it was an intense burden for them. And who knows, they were older too. I think the reason they adopted their son was because they couldn't have kids themselves. And when they passed away, who's going to take care of the son? And so I became very sensitized to these issues, because I could just see my neighbor, I won't say his name, but he was not going to be able to function that well in the world. His parents were already pretty old. What was going to happen? How stressful was that for that family? And so it's not something, if people want to criticize, they might say, "Oh, Steve, you're really insensitive, talking about screening embryos by their genomes." But I feel like I actually have some deep understanding of what life is like, for example, with intellectual disability, just because my next door neighbor was that way when I was growing up.
Right. Right. And it's, again, that I don't see how we could say there's anything wrong with knowing information and letting people make choices that are more informed. Because, yeah, obviously you're not telling people what to do based on that genetic profile. You're just giving them the information. So, okay, there's one thing I wanted to lean into as well. Perhaps we can just kind of briefly address it, and then I'll ask some of the other questions that I had planned to ask you. You mentioned something about schizophrenia and potentially having some type of ability to predict genetically if someone will have schizophrenia, which is very interesting to me. I've spent some time looking into schizophrenia pretty significantly, and of all the conditions in the DSM, my assessment is that for many of them, there may never be a genetic profile simply because of the, as people like to call it, heterogeneity within people with that condition. Many of the DSM criteria are so loose that there's no way that all of the people who fit those criteria will have the same neurological or genetic profile. So that makes it difficult. But schizophrenia, I do think, is one of the more clearly defined and easily recognizable conditions. So I can see how there could be a genetic profile that predicts that. So my question to you is, maybe, like, to what degree of certainty do you feel like you have in predicting it? And do you think that a person could have that genetic profile and be a sort of unique person? But, I don't, to whatever degree it's true that there's a connection between madness and genius, is there any possibility that that genetic profile could sort of have a positive outcome?
That's a great question. So I think it's good to elaborate on, for example, schizophrenia. It'll kind of illuminate really the nature of how genomic prediction works these days. Like how does the actual, statistical prediction of a heritable disease work? So, if you have access to a big data set, let's suppose you have hundreds of thousands of genomes from different people, and suppose you have a subpopulation in there that has been collected because they were diagnosed as schizophrenic, and maybe even have kind of maybe a severe case of schizophrenia. Okay, so imagine that you have, just making these numbers up, but suppose you have 20,000 genomes of people who have been diagnosed with schizophrenia, and then maybe 100,000 or 200,000 genomes of people with no schizophrenia diagnosis and seem to be completely healthy, psychologically healthy. Then what's done is that, using the fancy term now is AI or machine learning, you basically are analyzing those genotypes, trying to allow the computer to figure out which features of their DNA are correlated to being schizophrenic or anti-correlated to having had a schizophrenia diagnosis. And at the end, you have a predictor. Then you go to a completely different population of people that have not been used in any of the training. And the different population might even be people, and commonly is, people that were born in different decades, even on a different continent from the ones that were originally used in training. So you might have trained on some people in Europe, but you're testing it on some people in the United States or something like this. And then you just see if, conditional on having a high risk score, according to the computer, for schizophrenia, people with that risk score in the validation set, what is their probability of actually having a schizophrenia diagnosis? So you don't tell the computer who has this. It's all kind of, that information is not given to the computer, just each individual genotype. That's the only information that's given. And in the ideal case, you could even compare siblings. You could compare two brothers who were raised in the same family, one of which is high risk according to the predictor and the other one is not, and determine how often you called the correct sibling that actually has schizophrenia. So all this testing has been done. And a typical situation would be something like, if I know nothing about you, maybe you have a 1% risk of schizophrenia. I don't know the exact population prevalence, but I think it's in the low percents, maybe 1% or something.
Right.
So if I know nothing about you, the chances that I pick you out of a data set, the chances that you've been diagnosed as schizophrenic is maybe only about of order 1%. But for the subpopulation that the predictor says, these people are in the 99th or even higher risk for schizophrenia, there might be a 10%, 20%, 30% chance that in that subgroup, so conditional on the computer saying this person is an outlier for schizophrenia risk, the actual absolute probability that the person is schizophrenic could be 10 times or even 20, 30 times higher than in the general population. Mm-hmm. So we can never say with certainty, usually. I mean, for real outliers, sometimes you can almost say, like, for sure that woman is going to have breast cancer, and that we're almost at that level. But for the very best predictors, but for most of them, we can only tell you that this person has 10 times normal risk or 20 times normal risk. And so therefore, in a group of those people that's assembled, the fraction of them that have schizophrenia will just be enormously higher than in the general population. And that's what's meant by, quote, predicting schizophrenia risk. We're actually predicting the risk. We're not predicting that you have schizophrenia. We're just predicting that it's a little different with Down syndrome, because with Down syndrome, I would say, I'm not predicting the risk that the baby has Down syndrome. It will have Down syndrome because it's got
right.
Yeah. So we're not quite at that stage yet with these more complicated polygenic risk predictors, but we're getting there. There are cases where we could say, like, yeah, I'm 80-90% sure this woman is going to have heart disease.
Wow.
Or something like that. So yeah.
That's fascinating to me. And I would personally be very interested in, as this research unfolds and technology develops, when you are able to find, like you said, this subset of people who the computer says are at high risk of schizophrenia, and then let's say 20% of them have it, that would be extremely impressive to be able to compute and predict that. And I would also be very interested in the other 80% who the computer predicted would have it, and what their psychological profile might be, and if they might have some type of unique characteristics, but they've managed to stay functional.
Right. You might find that this particular set of people was predicted that they might end up having schizophrenia, but they didn't. And you might find it's because of certain environmental aspects of their upbringing or their family life, or something about their environment, not their genes, protected them against schizophrenia. So all of this is, if you just took a step back and you said, okay, in the 25th century, what is precision medicine going to look like, or precision psychology or psychiatry? It's got to look like that, right? You're going to, as one piece of information about your patient, you're going to want to have their DNA. You're maybe going to want to have some information about their upbringing, and et cetera, and some computer analysis. And, that's where we're headed.
That's a perfect segue too to the next questions I wanted to ask you, is essentially about the future. So maybe, this will be somewhat, like, not rapid fire necessarily, but I won't necessarily share all the thoughts that I'm sure will come to my mind to your responses. I'll just kind of ask a question, let you answer, and then we'll move on to the next one in an attempt to fit them all in.
Sure.
So my first question, and again, thinking relatively far into the future here, and so allowing ourselves to sound like we're science fictionist and things like that. First question is, what role do you think genetic testing and also genetic editing will play in curing conditions that are currently incurable?
So on the reproductive front, I think all these things will become more and more widespread. So IVF will become more widespread. Advanced genetic testing within IVF will become more widespread. And then as you go far enough in the future, editing will occur. So they will actually edit embryos, and it will become normalized after enough time. In terms of curing disease, so in the documentary, you saw, I think, a kid who had sickle cell anemia, and they were CRISPRing, I think, his blood cells to try to fix the issue. And I think you'll see that as a reality. So you'll find treatments where, by genetically editing the cells in your heart or in your prostate or in your eye, they'll be able to cure, perhaps, for example, a cancer or something that is potentially life-threatening.
Mm. I know I said I wouldn't respond too much, but this is actually something you might be able to help me understand better. So,
Yep.
as people may know, every cell in your body that has a nucleus contains your entire genome, but each cell only reads a certain segment of that genome, and the genes instruct what type of protein that cell manufactures.
Mm-hmm.
And so my question is, when you're making edits to the genome, like to a certain, let's say your eye, for example, to an attempt to correct a condition related to your eyes, does it work that CRISPR needs to be injected into that organ that contains the cells that reads those specific genes? But
Yes.
okay. So the genes,
So
yeah, please, you know where I'm
so
going.
the editing effect is specific to the region in which the CRISPR vector is injected.
Mm,
So in other words, it would be difficult for me to edit all the cells in your body. I mean, there's trillions of cells in your body. So if I know you have a specific problem, it might be heart disease, or it might be prostate cancer, or maybe some cancer in your eye, I can just inject in that local region. And if I have a highly effective CRISPR vector, it could potentially maybe get into 50% of the cells, or 75%, some fraction of the cells in that local region, and modify the DNA that's in just those cells. It wouldn't necessarily get into your neck or something like that, right? Or your hand. But if it just gets into the area where I know the problem exists, then I might cause a change in the functioning of those cells.
mm,
For example, I might be able to stop the cancer or slow down the cancer by editing the genome of the cancerous cells.
mm,
And so it's a localized treatment, and it might have to be done again.
right.
In other words, I might give you the injection, the cancer goes away, and then 5 years later, we detect it again, and they got to come in and CRISPR you again. So, but who knows? I mean, it's science fiction, right? But at the moment, actually, it's not entirely science fiction at the moment. I think there have been successful therapies of this sort already, but sort of niche. But we could imagine a future where customized CRISPR vectors are being injected to 150-year-old people, regularly to keep them alive, in the future. So,
Right.
yeah, this is not my main expertise. So I'm just speculating a little bit here.
Right.
So,
Yeah.
yeah.
That's the thing. I just want your perspective on this. I'm not necessarily holding you to being a complete expert on it, but I'm very just curious to hear that. So the next question is, again, thinking far into the future, let's say at least 50 years, let alone 100 or 200, how extreme could the social and cultural disparities become as a result of genetic editing and access to genetic testing and editing?
Huge. By the way, I should say, we have this habit of saying, like, oh, 100, like, okay, if we want to say something that's very dramatic, like it's very different from how things are done now, we would push it out 150 years. But actually, the rate of development of this stuff is super fast. So it's not going to take anywhere near that amount of time. I think by the end of our lives, we'll see a lot of these radical things already, deployed. In terms of social inequalities, it's huge, because you could imagine custom CRISPR treatments that, as I was saying, are keeping some 150-year-old guy alive. And what a privilege for this guy, this tech billionaire guy, to get to live 190 years when I only.
You know what? Can you hear me? Suddenly I can't hear you.
Yeah, we lost our connection there for a second.
There we
But
go.
I think we're good now.
Yeah. Yeah. So let's just, I know exactly where I was, like at the edge of my seat. So yeah, go ahead.
I was going to say back to my core competency on this. We can see, just as for any particular disease, whether it's heart disease or diabetes or whatever, we can now detect people who are in the high risk subpopulation, but we can also detect people who are in the low risk population. So there are people who are maybe, for example, 10 times less likely to have heart disease than a typical person, or 10 times less likely to have prostate cancer. So you can find high risk outliers and low risk outliers. And what's interesting about the genetic architectures of these diseases, just common diseases, the diseases most likely to kill you, um, is that they're largely disjoint. So it's different regions of your DNA that are affecting your diabetes risk than the ones that are affecting your prostate cancer risk.
Mm.
So if we did have 100% effective editing, I could independently take you, or take the embryo you before you were born, and make you low risk for prostate cancer, low risk for heart disease, low risk for diabetes, low risk for hypertension. I could do all of those things independently if I could make enough edits accurately.
Mm.
And so maybe that baby would grow to be 200 years old.
Mm. Right.
We just don't know. But I think the science is pretty strong that hypothetically a human with that genotype would just have much, much lower disease burden across all the common diseases than a typical person.
Right.
And of course, statistically, it's very unlikely that just a natural birth would produce an individual of that kind. Maybe they have, and maybe those are the people who live to be 125 or something.
Mm-hmm.
But I'm pretty sure that if the molecular jockeys do their thing, we will be able to engineer. I mean, we'll be capable of it. Whether we should is another question, but we will be capable as a civilization of engineering humans like that, certainly within 50 years. I would be surprised if it were, more than that.
So fascinating. It's such a fun time to be alive. And I agree, with what you said about how, we often think too far into the future when we're proposing science fiction hypotheses. And it's true. I think humans have a very difficult time grasping exponential growth.
Mm-hmm.
And that's certainly what is underway with everything we've been talking about. So if you had $1,000 and you had to place a bet right now on whether at any point in human evolution we would be talking about the potential of immortality, what would you bet?
Immortality is tough because you probably don't mean living forever, because eventually our sun is going to run out of fuel, right? So you'd have to solve some other problems as well, right? Yeah. So, yes. But radical life extension, I think, is not impossible. So, I mean, there are lots of animals that don't age the way that we do. Mm. And so I would not be surprised if radical life extension were possible. Hmm. I don't think that's an easy problem, though. So coming from physics, we're pretty hard-nosed and quantitative. So generally, the problems in medicine and biology that I would work on are ones that I think there's a very good chance we're going to be able to advance the ball. We're going to not necessarily fully solve, but we're going to get a good handle on it. Life extension is one of these things where it's very plausible to me that it's possible, but I'm not at all sure how long it'll take for us to develop those technologies. Mm. Other than this method that I just described, which is gene editing, embryos or babies so that they're low risk across all known diseases. I mean, that theoretically is possible.
So my next question is, what role do you think government has in all of this, in regulating and providing access, et cetera?
I think government, hopefully through a democratic process, has to get involved in this, because I don't think a Wild West is the right way to go. I think the inequality that we have right now is, bad enough. But I don't want billionaires having lifespans of 200 years, or billionaire kids having lifespans of 200 years, and other kids having lifespans of 60 years. I think that's terrible, right? So I think to some extent, there has to be some regulation and government involvement in this. I kind of think different countries might come to different conclusions on how to regulate it, or what should be allowed and not allowed. You could have a Wild West where some people travel to more laissez-faire countries to get genetic editing or various things done. But I don't think there's any. Well, I personally think government should get involved to some degree.
Yeah. And then what perhaps will be my last question. I feel comfortable saying it because I looked at your Twitter account and there's a picture of a couple kids, which I assume are your offspring. And so my question now is, again, kind of bringing up that idea that, sex is for recreation, science is for procreation. So if you are, or if you were in a position to be considering having another kid, would you ever consider, even if there were no fertility issues, doing it in vitro just so that you could get all this genetic testing and choose an embryo instead of the regular way?
Yeah, I would definitely consider it. So let's see. As far as that catchphrase, what did I say? Sex is for recreation and science is for procreation. Of course, that was, kind of a joke, but it's catchy. But it captures something, which is that, no, we're not going to stop having sex because we seem to like it. So that's never going to go away. But if there were a higher return, higher expected outcome method for producing your kid, which you might only do a few times, once, twice, 3 times in your life, produce a kid, why wouldn't you deploy all the technology possible? In the same way that we spend, if you calculate how much money some parents might spend for private schools for their kids or something, it totally dwarfs the cost of, a cycle of IVF or something like that. So I think, I personally, the benefits are, the real question is, okay, for people who do not have fertility issues and don't need to use IVF, are the benefits sufficiently high now that I would recommend that they actually do IVF, assuming they can afford it, just in case, because they might get a better outcome? I definitely think for some of my Silicon Valley, Wall Street friends for whom the money is, negligible, yeah, you would be crazy not to do it. You've got to maybe have to talk your wife into it. but yeah, you'd kind of be crazy not to do it, because if the economic cost of it is negligible to you, there really is a potential for better outcomes now. So people who are young, who start young, and we see this actually in our business, we sometimes see people coming in with 30 frozen embryos because they started when they're young. Like if you're a woman and you work at Apple, there's an egg freezing health benefit. Oh, you're in the Bay Area, right? So yeah. So, a lot of women in your neck of the woods who work in tech, they probably have an egg freezing health benefit, and they can just go and put away. And when you're young, one cycle of IVF, you might easily produce 10 or 15 eggs. So we've seen people come in with 30 embryos and for testing, and you can get a better outcome. You can just minimize, you can make sure you choose the one that's low risk in every category if you want.
And it's funny too, because I'm sure you've heard the phrase many times, are we playing God? And I'm, pretty agnostic. I'm not religious, but I can see a religious argument of, well, you should just go with the baby God made you. But what's interesting is if you're following that line of thinking, well, all of these embryos were sort of made by God in a sense. So there's really, to me, although I could see a lot of pushback, particularly from religious communities, specifically what we're talking about now with just IVF, genetic testing, and making a choice based on the highest likelihood of good outcomes. There's nothing anti-religious about it. There's nothing that goes against nature about it. And there's nothing where I don't know that we're thinking we're more powerful than we are. It's simply the scientific technology available to us.
I agree with you. I mean, I think very few people, although there certainly are people, and I respect their views, but most people who say are already in the pregnancy, they're already pregnant, and they find out that maybe it's a Downs baby. And that can be done just by a blood sample from the mother, right? You don't need to go do anything intrusive in the womb. You just test the blood of the mother, and there are enough DNA fragments of the child that are circulating in the mother's blood that we can tell, hey, that the baby you're carrying is going to be trisomy 21 or something. And most people would actually respect the woman who decides not to go through with that pregnancy. And of course, that's a much more drastic thing than to say that, oh, of these 30 embryos, of which I was only going to use 2 these are the ones I'm going to use, and the rest I'm going to donate to science. It's not really that drastic, because you can't even see these things with your naked eye at that point. But this woman's choosing to terminate her pregnancy based on some information she got, right? So, like, is that playing God? I mean, she's choosing not to have a baby, a thing that could become a baby, based on some information she got. Is that playing God? I think all of these questions are philosophically incredibly subtle. I think the people that have the most problem with it is if you believe life begins at conception. So the moment the sperm enters the egg and the embryo is fertilized, if you believe that those things all have souls, then to say I produced 30 souls and then I only used 2, that doesn't sound very good, right? So you should try to reduce the wastage, or just the number of embryos that are not used, because they all have souls. If you believe that, that's, I think, the logical next step for you. But most people, I think, don't believe necessarily that life begins at conception.
Right.
So, but there's an incredible spectrum, right? And if one of the things which I really get angry about is we're in America, which is primarily a Judeo-Christian country. In Asia, where there are quite a lot of Buddhists and Hindus, people have very different beliefs about the soul, reincarnation, etc., and they just have completely different feel for this. They actually don't feel nearly as concerned about these kinds of decisions in reproduction. And that whole perspective, which is actually the majority of the world's population, it's not the minority, it's actually the majority. There's actually more people who have those beliefs than the population of the United States or something. People here just can't accept the way that East Asians and South Asians feel about a lot of these things. They just don't think it's a ethical quandary to make some selection concerning your embryos or something, or even maybe an edit, I don't know. But anyway, but there's just incredible cultural insensitivity on the part of most Americans as to what other people in other parts of the world feel about these things.
Yes, that's a very interesting point. And I suppose for the people who do believe life begins at conception, then who knows, maybe they'd be more open to editing genetics.
Exactly. No, exactly. Yeah, no, Nick, that's a great point. If you believe that the soul enters the creature the moment that conception happens, then you should do your damnedest to, if you need to help this little soul trapped in these few cells, if you need to go in and edit to help that soul have a better time on Earth, why wouldn't you do it? I mean, it's just amazing the range of beliefs that you can have about all this stuff.
Yes. And perhaps I'll just share a fun fact for people watching and listening that I discovered just a few years ago that I thought was fascinating, which is that when you have a frozen embryo, it's actually very hard to know when the sort of expiration date on that is. And it's much longer than people would think. And and there was, I believe, the longest time an embryo has ever been frozen and then implanted and was born was 26 years.
Yes.
And a 25 or 26-year-old woman gave birth to that child. So this mother and her child were conceived in basically the same year, but the embryo of the child was frozen. And I mean, it's frozen in time, quite literally. There's no degradation whatsoever. So that's just fascinating to me.
I've been in clinics where, they have very fancy, these embryos are frozen in something called liquid nitrogen. So it's extremely cold. It's actually, liquid nitrogen is a substance which is used a lot actually in physics labs, because we often need like super cold environments to test certain things. And, you walk through the storage area and you just see vat after vat of, they're called cryostats. They're things which are held at a very low constant temperature. And there could be thousands and thousands of frozen embryos in one of these facilities. And I think actually with good quality control and monitoring of the temperature, 26 years is not anything like the limit. You could probably, I would not be surprised if you could freeze an embryo for hundreds of years and then use it after that.
That's so fascinating. Wow. Well, you've generously given me an hour and 15 minutes of your time, and I'm so grateful. This was so thought-provoking for me. I think we ended up focusing a little bit more on genetic testing than genetic editing, but that felt perfectly appropriate and relevant. And I thank everyone who's listening and watching at this point for watching all the way through. I'm glad it was intriguing enough. And yeah, and again, this is the Psychology Is podcast, where people are, psychology is. That's our kind of phrase. And we talk about all kinds of different things. And I'm really glad we were able to bring in this topic because, again, it's just personally fascinating to me. And I just think it's one of the most cutting edge conversations to be having. So thank you so much.
It's been a pleasure, Nick. Good luck with your podcast.
Thank you. Good luck with Genomic Prediction and all of your endeavors.
All right. Thanks. All right. Have a great day.
You too.
If you like this video and you want to see more, please subscribe.